Now, as we know that lipid digestion in the duodenum produces free fatty acids, free cholesterol, and 2-monoacylglycerol. These, along with bile salts and fat-soluble vitamins A, D, E, and K, form micelles, which are small clusters of lipids that are soluble in water. Therefore, they can move easily through the aqueous environment of the intestine. There is an unstirred water layer that surrounds the brush border of the enterocytes. This layer consists of still and stagnant water that creates a barrier between the intestinal lumen and the enterocytes. It also contains a layer of mucus secreted by the goblet cells. This layer allows the selective uptake of nutrients by the cells. This is essential because some substances may be harmful and not all are beneficial. It acts as a filter, preventing the absorption of larger molecules and bacteria. The hydrophilic surface of the micelles facilitates the transport of hydrophobic lipids through the unstirred water layer to the brush border of the enterocytes where they are absorbed. Short and medium chain fatty acids are water soluble, so they do not require micelles for their transport. Absorption of bile salts occur in the terminal ileum and rest is lost in feces. Cholesterol, however, is poorly absorbed from the enterocytes. Now the lipids, once entering the enterocytes, migrates towards the smooth endoplasmic reticulum where resynthesis occurs. However, short and medium chain fatty acids are not re-esterified and are transported across the cell membrane and into the portal vein, which carries them to the liver where they are metabolized and used for energy production. Long-chain fatty acids are first converted into their activated form, fatty acyl coenzyme A. Next, an enzyme monoacylglycerol acyltransferase catalyzes the transfer of a fatty acid from a fatty acyl coenzyme A molecule to the monoacylglycerol molecule, forming a diacylglycerol molecule. Another enzyme, diacylglycerol acyltransferase, catalyzes the transfer of an acyl group from a fatty acyl coenzyme A molecule to the diacylglycerol molecule, forming a triglyceride molecule. Lysophospholipase are a type of phospholipid that have only one fatty acid chain attached to the glycerol backbone, as opposed to two in the case of regular phospholipids. Reacylation of lysophospholipase is the process by which lysophospholipids are converted back into regular phospholipids by an enzyme called lysophosphatidylcholine acyltransferase. The reacylation of cholesterol is catalyzed by an enzyme, acyl coenzyme A cholesterol acyltransferase. This forms a cholesteryl ester molecule and releases coenzyme A. Steatorrhea is a medical condition in which there is an excessive amount of fat in the feces. This results in pale, bulky, and foul-smelling stools. Steatorrhea is caused by malabsorption of fat in the small intestine, which can be due to a variety of medical conditions such as pancreatic insufficiency, in which the pancreas doesn't produce enough enzymes that are necessary for the digestion and absorption of fat in the small intestine. Celiac disease, in which the lining of the small intestine is damaged, leading to malabsorption of the nutrients including fat. Crohn's disease, which is an inflammatory bowel disease that affects the digestive tract. Bile duct obstruction. If the bile ducts that carry bile from the liver to the small intestine become blocked, the absorption of fat is affected. Other causes of steatorrhea include bacterial overgrowth in the small intestine, certain medications, and diseases that affect the lining of the small intestine. Now we will discuss the secretion and use by the tissues in the next video. Thanks.